Thank you very much for the invite and the introduction and the choice of paper. Let me know if you have any problems seeing my screen or mouse or anything. So this is the paper that I'll be discussing. It was published this year in Neuropsychopharmacology, and really great thanks to all the authors, but particularly Sara Costi, who pretty much ran and collected all the data for these projects, and I was just there to analyze it. So big shout out to Sara. So before I dive into this paper, I'm just going to take you back to a preclinical paper that inspired a lot of the analysis by a group at the other place, Angela Roberts' group. They studied the role of the subgenual cingulate cortex in non-human primates in marmosets, looked at its role in proxies of anhedonia and anxiety, and the influence of ketamine on its activity. So just to briefly, as a bit of background, take you through that, starting with the subgenual anterior cingulate cortex. What is it? It's a region under the genu of the corpus callosum. It's a cortical region. It's involved with a range of different cognitive and clinical states, mainly negative mood, sadness, motivational tone, arousal for anticipating rewards and punishments. A few different types of studies have shown grey matter loss in this region in depression, both from structural MRI and postmortem studies. However, it seems to paradoxically have elevated activity, both metabolic and bold activity during fMRI. And its activity is actually dampened by antidepressants and deep brain stimulation. So this seems to be an important and interesting region in depression and related symptoms. So this primate study actually overactivated the subgenual cingulate cortex in 5 marmosets and observed the expected anxious and depressed-like phenotypes in these primates. So they saw a reduction in anticipatory arousal, and this was measured as mean arterial blood pressure during anticipation for rewards, and also a dampening in head jerks, a behavioral measure of anticipation for rewards when this region is overactivated, but no difference in consumption of reward. So something about its role in anticipation of reward. They also had this measure of anxiety, showing that overactivation of this region increased behavioral measures, a range of measures of anxiety. They have a human come in and stare at the monkey for a while, and they measure their behavioral response as various different measures of anxiety. So sort of replicating what we already expect in the overactivation of this region. And this was actually overactivation via an increase in presynaptic glutamate. And that caused reduced anticipatory arousal and reduced motivation, and all these a variety of different depressive and anxious phenotypes in marmosets. And interestingly, when they applied intramuscular ketamine, a single intramuscular ketamine infusion, it seemed to normalize these depressive and anxious phenotypes. So it returned the anticipatory physiological arousal measures when they're anticipating reward, and the behavioral measures as well. It also normalized the hyperactivity of the subgenual cingulate cortex. So it seemed that intramuscular ketamine in these marmosets seemed to normalize not only the behavioral phenotype, but also this neural phenotype of subgenual cingulate hyperactivation. And interestingly, they compared an SSRI, and that didn't have the same effect. So just going back a little bit further, why is this region hyperactive in the first place? There's quite a wealth now of evidence that indicates that excessive stress-related hippocampal glutamate outputs to the subgenual cingulate cortex is responsible, or at least related to, this increase in subgenual anterior cingulate activity. And there's a range of preclinical and clinical studies to show this, including a study just showing that there is this increased connectivity between the hippocampus and subgenual cingulate cortex in patients with depression, and that is associated with severity and the illness duration. And actually the same group that worked in the marmosets showed a similar pattern where this connectivity, this pathway from hippocampus to the subgenual cingulate cortex was important for correct punishment avoidance. And actually, if you increase the glutamate in the subgenual cingulate cortex coming from these hippocampal projections, actually increases punishment avoidance, which they suggest is a marker of a depressive phenotype in those marmosets. So that sort of gives us a clear mechanistic model that we can test in patients with depression in humans. So just a brief summary of that background and the rationale for the analyses that we did. We know from various studies that heightened stress-induced hippocampal glutamatergic output to the subgenual cingulate is often causal of depressive symptoms and is observed in patients with depression. And that increased subgenual cingulate activity is associated with a range of measures related to depressive symptoms and anxious symptoms, and that hyperactivity seems to be normalized by ketamine. So now just moving on to our study, we're trying to demonstrate and replicate all of these things in patients from a series of studies. So the first one was funded by an R21 grant for James Murrough, and we recruited, and Sara Costi did a lot of this work, so she can speak to procedural and clinical information about the recruitment and all the procedures. So depressed patients were recruited and they were diagnosed based on the DSM SCID. They had exclusions of comorbid psychiatric disorders, excluding anxiety disorders. They were free of antidepressant or other psychotropic medication. And here are some of the demographics, about 28 patients and 20 controls. The main measure of depression was the MADRS self-report scale. And then we used the TEPS scale to measure different facets of anhedonia, both anticipatory pleasure or anhedonia and consummation, and an anxiety scale as well. We used the STIXA. So that's the population, and we scanned these patients and we had them do a task during scanning and also a resting state scan. And during the task, it was a very simple task. It's similar to the monetary incentive delay task, or MID, if you're familiar with it, but it's very straightforward. Patients during this task, they view a cue. It could be 0 cents, 10 cents, or 50 cents. And then they do a very brief cognitive task where they just have to respond to the middle letter in a row of letters, and sometimes they're congruent and sometimes they're incongruent. They make their response and then they get feedback, either positive feedback or negative feedback, or neutral feedback. And so this is really what we focused on, this subgenual cingulate reactivity to this main part, which is the incentive feedback. And we also looked at the connectivity between the hippocampus and the subgenual cingulate cortex. And so what we found was we were actually able to replicate a number of studies and what we would expect, seeing hyperactivation of the subgenual cingulate cortex in patients with depression compared to controls, both when receiving positive feedback and negative feedback. So it's taking it a step further than what we already know, because there's not much on task-based subgenual cingulate cortex activation. It's mostly during rest. And we were actually also able to dissociate the hyperactivity such that hyperactivity of the subgenual cingulate to positive information was more associated with anticipatory anhedonia, whereas hyperactivity to negative information was more associated with anxiety. And that's across patients and controls. And then aiming to replicate the increased connectivity between the hippocampus and the subgenual cingulate cortex was possible. So in the same subjects, we saw that hyperconnectivity. And interestingly, we did see that a relationship between that increased connectivity between those regions and the task-based subgenual cingulate hyperactivation only to the positive information rather than the negative information that they were receiving. And then following on, we had a second study funded by the Doris Duke Charitable Foundation. This is the study I just explained, and this is the next study, a smaller N, so only 16 patients, slightly higher MADRS scores, which reduced after ketamine, and also slightly higher anhedonia scores, which reduced again after ketamine. And I point you towards Sara Costi's recent paper, which outlines this, this study in much more detail. But for this analysis, we just analyzed the data for a single intravenous ketamine infusion. And we had pre and post ketamine MRI scans using the same task around 2 weeks to 5 days before and after the infusion. And what we found using the same task was that ketamine within subjects seemed to reduce the subgenual cingulate hyperactivation, again, to the positive information, not to the negative. And remember, this positive hyperactivity to the positive information seemed to be more related to anhedonia symptoms than anxiety symptoms. And we did see a relationship, but this is a small n, so I wouldn't take too much from it. But we did see a relationship between the baseline subgenual cingulate hyperactivity to positive information and a greater improvement in anhedonia. So not only did ketamine reduce or maybe normalize the subgenual cingulate hyperactivity, but there might be some prediction of anhedonic improvement as well. So we're just coming up to the end. There was just one final exploratory analysis that I was interested in doing, which was trying to tease apart this large subgenual region. In non-human primates, there's actually cellular distinction between like the anterior and posterior subdivisions of this region. So I just did a split based on some anatomical boundaries to see if there was any difference between the anterior region and the posterior region, with the posterior region being more akin to area 25, whereas the anterior region is more akin to area 32. And we did see a small dissociation where area 25 seemed to be more related to the hyperactivity in the positive domain, whereas the area 32 was more related to hyperactivity in the negative domain. And as expected, just based on that, this hyperactivity in the positive domain, again, was associated with anhedonia and not anxiety. And whereas area 32 hyperactivity to negative information was more associated with anxiety and not to anhedonia. So, I mean, this is kind of preliminary, but an interesting anatomical distinction and definitely warrants replication and further study. So yeah, judging from that and some of the background that I presented, the current working model based on this is that there might be some excessive stress-related glutamatergic projections from the hippocampus to the subgenual cingulate, which leads to subgenual cingulate hyperactivity. This hyperactivation to positive information is more associated with anhedonia, whereas hyperactivation to negative information is associated with anxiety. And ketamine seems to specifically blunt the hyperactivation to positive information, and symptom on a symptom basis, it does reduce anhedonia, but it also reduces anxiety. So there might be another neurological reason for that. So yeah. Thank you to everyone for listening and to all the co-authors, especially Sara and James.
Fabulous. Thank you very much. That's a real triumph. I think it's really, really interesting. I just encourage people to use the Q&A, as I mentioned before, for their questions. I have the great privilege of asking the first one, and that is, do you think that there's any relationship between this, just at a clinical level, between this hippocampal to subgenual hyperactivation and rumination?
Potentially. Potentially. We just published a paper in collaboration with a group at Sinai, doing a whole brain network analysis using high field MRI with good spatial specificity to be able to answer some of these questions. And they actually found that it was a pathway from the precuneus to a medial prefrontal region. It was more OFC than subgenual cingulate cortex. was associated with negative rumination in a good sample of depressed patients. So these kinds of whole brain data-driven analyses will be the best at determining that. And we know the default mode network, which does sort of extend to subgenual and really includes a lot of the medial wall of the frontal cortex and the precuneus and the PCC area. We know that network is involved with rumination. And so this recent paper that we published suggests something about the directionality of that, PCC2 prefrontal or medial, ventromedial prefrontal. As far as the hip, we didn't see much coming from the hippocampus, but I think maybe we should have done specific analyses just looking at that pathway and exploring that a bit more.
Okay, and then I had one other question, which is about, do you think this is in any sense related to the disappointment, the sort of the anti-reward center of the lateral habenula? Do you think it's got anything to do with that?
So I've been looking in like human MRI for habenular projections, and there's definitely a lot of preclinical evidence suggesting that habenular projections to the medial prefrontal cortex in rodents is associated with punishment processing, negative processing, but it seems more related to projections to the VTA.
Yeah.
I've not been able to replicate that with the current resolution that we have, which is not really good enough probably to look at habenula, and definitely not good enough to look at lateral habenula. But yeah, I think there's definitely something that we should keep trying to specify and to analyze.
So just for somebody who's not really a neuroanatomist, am I right in thinking the lateral habenula is more involved in sort of disappointment, if I can put it like that? But this, how does that relate to this anticipatory anhedonia?
So the lateral habenula, like you say, it becomes activated when you're disappointed, when you receive some punishment, or when you don't receive a reward. So it's like the antithesis to the ventral tegmental area, which responds when you anticipate a reward or you receive a reward. So, I mean, without any evidence in my mind, I would predict that the habenula would be more active in that disappointment phase when they're receiving or learning that they've received something negative or they've not received something positive. So that would be more in the receipt phase of interacting with a reward or punishment rather than the anticipation phase. But I think that needs to probably be tested and confirmed.
Okay, so we've got a question from Tiago Gil. At the SKP conference, I presented our hypothesis that depression is one form of seizure activity in specific brain areas. This study corroborates this hypothesis. Can I hear your thoughts on calling it seizures or hyperactivity? What's the difference in physiology?
Great question. So seizures, if I'm not a neurologist, but seizures, if I understand, a hyperelectrical activity of specific brain regions. And what we're studying is blood oxygenation changes, which is quite a few steps removed from actually measuring electrical activity of the brain. However, having said that, there are some studies that show that increased electrical activity in the brain is associated with an increase in our measure, in this bold measure or this oxygenation measure. However, I do think that the increase in electrical activity produced by a seizure might be on a different scale to the changes that we see in the bold signal during one of these fMRI scans. But without doing simultaneous EEG and fMRI, I don't think, I'm not sure if I could say exactly.
So we haven't got any other, oh yes, we've got a couple of other Q&As, they've come through. So is this yet another example of a mechanism of action for ketamine, or does this study suggest that stimulating new dendritic synaptic growth is not ketamine's main mechanism of action? So how does it relate to?
I don't know. I don't think this study could say one way or the other. I do think that just because we're reducing activity in one part of the brain with ketamine doesn't mean that reduction of activity is reduced to is related to reduced structural proliferation or really any structural changes. There could still be new dendritic and synaptic growth of inhibitory circuits or inhibitory control of the subgenual cingulate cortex. That's probably a question for follow-up analyses, but I think, yeah, you probably have to do some histochemistry to answer that.
There's a good question from Laurie Calabresi. Although the scans were done up to 14 days after ketamine, what was the average number of days after ketamine to scanning?
Yeah, good question. So the 14 days was actually the variability around the first scan. So it could have been up to 14 days between the first scan and the infusion of ketamine, but the post ketamine scan was a variability of up to 5 days. I don't remember the average number of days, but I can find out and get back to you. But that's definitely very important. Yeah.
Presumably that would be related, but I mean, is that related in any way to the possibility of synaptic growth, dendritic synaptic growth, the time course of that?
Potentially. I will say that in an analysis, controlling for how many days after scan, it didn't change the results. So at least for these specific findings, it didn't make a difference. But definitely, I think with larger sample sizes and more studies done on ketamine and brain imaging, we'll be able to answer some of these questions. You know, at what point do the clinical symptoms take effect? At what point do the neural changes take effect? And this is going to vary on an individual basis. And I know Sarah's doing a lot of work and all of you are doing a lot of work to answer these questions. So I think it's beyond the scope of the current study, but I'm looking forward to more data being available to answer that question on the timeline of changes.
Sure. Were the subjects on antidepressants, the patients? No,
they weren't.
The patients were antidepressant free, were they?
I believe so. Sarah?
Okay.
Confirm?
Yeah. I confirm. Laura, thank you for the talk. It's been like great attending your talk as usual. I just have a question on one of the last slides you showed about the different area of the subgenual cingulate. And I think you were referring to like study one, like in MDD versus healthy. But did you run like similar analysis also in the group pre and post ketamine, or was maybe the sample size too small, or what would you expect? Any difference in the ketamine action between these two regions, or it's difficult to say?
Yes, I mean, important question. I did run the analysis. I can't actually remember off the top of my head what the findings were, which probably tells me that there wasn't much, and probably, like you say, related to the small sample size, a very small sample size really. And yeah, and unfortunately the imaging procedures weren't as strong as this larger study, so that to be able to spatially differentiate between those signals would probably be difficult as well. But I'm gonna follow up on that and double check, because given this, you know, because of the relationship between ketamine and blunting the more anhedonia related neural correlates, I would expect that maybe this region would be more affected by ketamine compared to this region, just based on the previous study. But I'm going to double check on that and get back to you.
Thank you.
So I mean, it's a simple. I mean, maybe it's a stupid question, but why is hyperactivity associated with anhedonia?
Yeah, no stupid questions. I think we need to do a lot of neuroscience to figure that out, but this region has been associated with negative mood and sadness. If you show people sad images or play a sad song, the subgenual cingulate cortex becomes active. So I don't really, I mean, the point that it is hyperactive to both positive and negative information is strange, because you'd expect it would just be hyperactive to negative. But maybe there's some aberrant recruitment of negative neural systems that that mediate negative information. There's some aberrant recruitment of that during positive information in depressed patients. Would be one way of interpreting this data.
Yeah, so sort of having a glass half empty view of the world. Yeah.
Yeah, maybe.
I understand all that.
It's just depressive realism.
Yeah. Okay, good. And so hard to tease out whether it's effect of the ketamine or of its antidepressant effect.
Yeah, I think, well, actually, maybe not, because these findings, at least the relationship between, you're right, but at least the relationship between the subgenual cortex hyperactivity and the symptoms, we saw that same trend in healthy controls as well. So there's a little bit of variability in presentation of anhedonia and anxiety in the healthy controls, and so we saw a relationship between the hyperactivity and that variability as well. So, you know, they're not showing any clinical symptoms, it's just some inherent variability. So maybe that says that it's not related to strong clinical presentations, and maybe it's just related to the neural function, but I don't know.
That's a great note to end on. Thank you.